The granulocytes are the neutrophils, eosinophils, and basophils. You need to know your different cell type. You need to know that neutrophils are the most abundant. You need to know the function of each cell. So the neutrophil is a phagocyte. The eosinophil is also a phagocyte, but it is going to increase when there is an allergy or when there's a parasitic infection. Basophils are not phagocytes. They enter the damaged tissue and release histamine. What histamine is going to do is promote inflammation. Inflammation is when tissue becomes red, warm, sore, or painful, and swollen. And this is because of chemicals, the histamine in particular, that causes the capillaries to get leaky. And this allows more flow of particles, nutrients, oxygen, white blood cells to the area. So basophils promote inflammation. The agranular leukocytes or agranulocytes are the monocytes and the lymphocytes. After neutrophils, lymphocytes are most abundant. The monocytes are macrophages, are phagocytes, and they become macrophages. So when it's in the blood, it's called a monocyte. But once it escapes from the blood and enters the tissue, it will be called a macrophage. And throughout anatomy and physiology, we'll hear about these macrophages. They are just monocytes that have left the blood, and they act as phagocytes. Lymphocytes have got a variety of functions, and we will say that they provide immunity against pathogens. It's kind of interesting if you look at how long these cells last. Monocytes can survive for months or longer. Lymphocytes can last for decades. And what they're going to do is remember the infection so that you can fight that infection more quickly the second time you're exposed to it. Which white blood cell increases in abundance in response to allergies? Eosinophil. Which white blood cell is the most abundant type of agranulocyte? Lymphocyte. The clotting response is a complex cascade of events that reduces blood loss. In this section, we're going to discuss the mechanisms that control blood loss after an injury and describe the sequences responsible for blood clotting. Well, hemostasis is blood stopping, and the process responsible for stopping blood loss has three different phases. The first is the vascular phase, the second is the platelet phase, and the third is the coagulation phase. The vascular phase lasts about 30 minutes, and once the blood vessel has been damaged, and here they're just showing that a knife has cut into the blood vessel, there is a vascular spasm. So our blood vessels have smooth muscle in them, and this smooth muscle will contract. This happens when the exposed cut endothelium comes in contact with the blood. When there is contact, those endothelial cells are going to release chemicals and local hormones called endothelins. The smooth muscle contracts, that's a vascular spasm, and the division of other cells are stimulated for repair. The second phase is the platelet phase. Platelets are sticky, and so the platelet phase begins with the attachment of these sticky platelets to the endothelial surface. So endothelium is that special word that represents the inner lining of our blood vessels. It is epithelium, and it is simple squamous epithelium, but when it's in our blood vessels, it's called endothelium. And so the platelets adhere to that because it's been cut through and it's exposed, and start the formation of a clot. These platelets will then release other chemicals. One is adenosine diphosphate that stimulates platelet grouping, aggregation, and secretion. And then chemicals that stimulate vascular spasms will be released. Platelet factors are proteins that will be involved in clotting. And then platelet-derived growth factors promote vessel repair. You do not need to know these chemicals. Calcium ions are also required for the clotting process. You do need to know that calcium is important for clotting. The third phase is the coagulation phase. So here in this picture you can see that it looks like some sort of thread is capturing these red blood cells, and that's exactly what it is. This starts about 30 seconds or more after the damage, and there is a sequence of events or a cascade of events that will convert that circulating fibrinogen, that's a plasma protein, and it's soluble, but the cascade of events will cause it to become insoluble, and so these fibrin threads come out of solution and catch the red blood cells to form a clot. The coagulation phase also involves some other enzymes, and calcium's required, and look at this, 11 different proteins, and they are numbered with Roman numerals 1 through 11. So there's quite a lot going on in this coagulation phase of hemostasis, and basically we skip it all. Well, there are two pathways that lead to the common pathway, and that is the extrinsic and intrinsic pathway. We'll look at a diagram of these two pathways, but their names are extrinsic and intrinsic, and basically you just need to know that, their names. There's the intrinsic, but we'll look at that in a second. And both of those will lead to the common pathway. And that common pathway is going to lead to the conversion of the fibrinogen protein in the blood to fibrin threads that will trap the red blood cells. So, basically, the enzyme thrombin, well, there's a lot going on to produce that enzyme, but the enzyme thrombin converts fibrinogen to fibrin. Clot retraction, the red blood cells and platelets stick to the fibrin threads. The platelets contract to form a tighter clot and pull the edges together, and this will go on for about an hour. So this is the diagram of the extrinsic pathway and the intrinsic pathway, and both of them are leading to that common pathway. So in our blood we have all of these factors, all these Roman numerals 1 through 11, and each factor has a different function. And so they will all work separately to result in the formation of soluble or insoluble fibrin. The thrombin, that enzyme thrombin, will convert fibrinogen to fibrin. Fibrinolysis is the process of the clot dissolving. And we don't need to know how that takes place. Which of the following is not a function of platelets? Expose collagen fibers. What is the function of the endothelians? vascular spasm. Which ion is essential to the activation of some clotting factors? Calcium. Which enzyme converts fibrinogen into fibrin threads? Thrombin. Blood disorders can be classified by their origins and the changes in blood characteristics. So, first you've got to draw some blood. The phlebotomist will draw the blood. It is a process called venipuncture, and you're withdrawing from the vein. There's too much pressure in the artery to withdraw from the artery. So you're going to withdraw from a superficial vein. The superficial veins are easy to locate. The vein walls are thinner than artery walls, so it's easier to get the needle through. And venous pressure is low, so the vein will seal quickly. So we'll look at some nutritional blood disorders. Iron deficiency anemia is not enough iron in the diet. The result is that the red blood cells are very small, microcytic. Micro means small, and they're going to transport less oxygen. This is more common in women than men. Women have fewer iron reserves than men. Pernicious anemia is a vitamin B12 deficiency. You can get vitamin B12 shots to prevent this, but with pernicious anemia, fewer red blood cells are produced, but they're large. They're macrocytic. And this can be from a lack of intrinsic factor. We produce intrinsic factor in our stomach, and that intrinsic factor is needed for vitamin B12 absorption. If you don't have intrinsic factor, you'll be low in vitamin B12. Alcoholics are often low in vitamin B12. Calcium and vitamin K deficiencies can cause problems. Calcium is required for clotting, and vitamin K is required by the liver to make the clotting factors. A congenital blood disorder is sickle cell disease. It's a genetic mutation. It affects one amino acid in each of the beta chains of hemoglobin. And under low oxygen condition, the red blood cell will take on that sickle shape. So those purple cells there are misshapen red blood cells. They can easily get stuck in joints and capillaries and reduce blood flow. So to have sickle cell anemia, you get one copy from both your mom and the dad. If you have only one copy instead of both copies, then you have this sickling trait. Those people will spend quite a bit of time in the hospital. If you have sickle cell anemia, you will die. Hemophilia is an inherited bleeding disorder. It's missing one of those factors in the blood. Now we can produce those factors through genetic recombination. But there's quite a few different forms of hemophilia. It affects one person in 10,000. And the gene is found on the X chromosome, so males have it more often than females. And a thalassemia is a type of anemia found in Middle Eastern people, and they're unable to produce normal hemoglobin protein subunits. So it can be mild or severe depending on how many subunits are abnormal. Blood infections. Bacteremia is bacteria circulating in the blood but not multiplying. Viremia is viruses circulating in the blood but not multiplying. Sepsis is a widespread infection of the body. Septicemia is blood poisoning. The pathogen is present, multiplying in the blood and rapidly spreading.